to the next topic, which is looking at human microbe interaction. And I can tell you right now, this slide set's going to get updated probably more than once because I'm going through the material every year. I have to brush up on. I've been saving a lot of topics. Now I need to put them into the PowerPoint because this information is changing so rapidly. There's so much new information pouring into the published literature. It's really challenging to stay up on it for that person especially. So, but I will be adding into it. This is a really, really exciting area of research. I'm so happy to see all the funding that is getting put toward this effort. And so there's been historically all kinds of effort to investigate all the pathogens that attack humans and other animals. But now there's this big push for us to try to understand the normal microbes that just hang out with us. And the more they look, the more astounding the results are. And I actually think not only is it super exciting, but also very hopeful. There's so many good studies coming out with good data to show that we have far more control on our health than we ever thought possible. And if we just have some basic understanding of this incredible dynamic between yourself and your microbes, it literally could change your life in a very positive way in terms of your overall health and well-being. So there's been a lot of shifting going on with this idea of humans and their associated microbes. So for example, not too long ago, the idea was this, and you still see this out in the literature. They're not posted yet? Really? Okay, that's weird. Just before I did that. Okay, let me pause for a second and get in there and do that. I'm going crazy. More coffee. All right, microbe, human microbe. Like I said, these will get updated, but we can start with this. All right, let me So not too long ago, and you'll see this out in the literature to this day, and people still quote this, and they're wrong. But it's a very nice thing to think about. It's just a little off. So this was the previous population. They say the adult human has about 1 times 10 to 14 cells that make up the human body. And then this idea came about that they said, oh, we're really only about 10% of you are in eukaryotic cells. And the other 90% are the microbes. That was proven to be not true. But it sounds pretty, it really kind of hits you. What? 90% of me is not me? It's very stunning data. And so people, the common media ran with that, Times, Newsweek, all them, they go like, oh yeah, we still see this a lot. I see it a lot on YouTube videos. But please, if you're interacting with people and they start spouting that off, correct them. It's still true that we are more microbes than we are eukaryotic cells. That's still true. So the more careful study that they did, they took the standard man and weighing 70 kilograms, and they were running the data on that. They said for every 1 human cell, there's 1.3 microbes, microbial cells. So 1.3 microbes to 1, 1.3 to 1 is the ratio. So it's not 90 to 1, 9 to 1. And so you can still say that we're more microbial than human. That's accurate. So what had happened with that previous hypothesis, there was a couple of scientists that, it was literally like the back of the napkin type calculation where they were, having lunch together, and they kind of did this math calculation, and somehow that blossomed into this data that everyone kept quoting. And then they said, we should probably verify that. So then they actually published a study with good data, and that's where that 1.3 to 1 idea is coming from. So I love this quote taken from a published paper. The human body represents a scaffold upon which multitudes of mutualistic and symbiotic species build residence, creating a diverse ecosystem composed of members of 5 of the 6 kingdoms of life. So we really are symbiotic. We really are an ecosystem. And we have very intimate interactions between our body in pretty much any area of our body and with the microbes that coexist in those areas. And so it is this, again, so much of the shifting information. When I first took microbiology a while ago, we were told there are certain parts of the body that are sterile. There are no microbes there. And now we realize that that's not necessarily the case. This is probably one of the more contentious body sites, the womb, the uterus, thought that those areas were sterile. Some people are finding that you can find microbes that aren't pathogenic hanging out in the placental tissue. They're there. Some have sampled the amniotic fluid and found low population of bacteria there that, again, are not pathogenic and seem to be just living there and living their life there. Other scientists have been coming back and saying, no, you're wrong. And they're saying that was a experimental error. That's, you got contamination of your sample. So this is a hot topic right now. There's a lot of debate going on. I read the studies in the first bullet point where they were finding, and to me, the experimental design and the method of capture of these organisms all looked legit. Sure, there could have been a mistake, I suppose, that they didn't report on because they didn't realize they were doing it. But I tend to think that we do have colonization there of beneficial microbes. But so this story still needs to be fleshed out. Certainly, right upon birth, this is not contentious. We know that as soon as the mother goes into labor, that baby is starting to get colonized as soon as they start working their way down the birth canal, the vaginal canal. And so they've actually done some studies looking at the difference between if you were born through vaginal delivery or C-section. It makes a difference with your microbiome early in life in particular. They've sampled, and so the way they've been sampling babies, obviously they can't be super intrusive with that, so they'll take fecal samples. They'll analyze the feces and they'll look, and they can see just through fecal sampling, you can see a definite difference in the gut microbiome depending on if it's a vaginal versus a C-section. And so I had one of each. My 2 boys, first one was C-section, second was vaginal, and I wish I would have taken samples way back then and ran the test, but they have shown that there's a definite difference. And then as the baby starts to grow and develop, you'll start to see kind of like a succession of microbes over time. But by the time you're an adult, you are all unique. You have a very unique signature of microbes that make you. We all have pretty much the same phylum of microbes showing up, but there's some nuances that make you very unique compared to other people. So certainly you can get vertical transmission of microbes when you think about when you were born. Of course, anything and anyone you interacted with was an opportunity for microbes to transfer to you. If you were breastfed, there are studies done with breast tissue and breast milk that there are microbes in the breast tissue in the breast milk that get transferred to the baby. Certainly, all of the skin tissues, though, they say that it's typical that the mother has the strongest influence initially, which makes sense because the mother is the one that is breastfeeding and handling the baby a lot. Then there's a lot of transfer, skin to skin transfer as well. So lots of opportunities there. But then, just thinking about the family that baby is raised in, will have, other people in the family, the father, the kid, other kids, potentially the animals, the pets, the messy house. So very much And back when my kids were really young and, you worry about them getting sick with some pathogen, New York Times put out an article on that. There's a picture of a baby sitting in a mud puddle and just splashes of mud all over the baby, and it's got a gob of mud and it's bringing it to its mouth. And the title was something like "Let them eat dirt" or something like that. But the whole article was about this idea that you don't, within reason, I mean, you have to be worried about some things, certainly, but within reason, it's okay to get dirty and to, play around in the soil and all of that. And it's actually helping that child, and you shouldn't be so freaked out about it. And they also made the point that a super clean house isn't necessary. I was very happy to hear that because my house to this day is not super clean. So that's another reason why I'm like, yeah, dust bunnies, who cares? It's helping my kids with their immune. So they have this certainly very potent interaction in the home. Think about human babies, and then as that child progresses into their life and shifts into adult phase, they do see a succession of microbes. And also, like I said, there can be some commonalities across the human species that have been sampled, but they can now, with better technology, they're certainly able to point out that there's some unique things going on with each person. So these 4 phyla I would like you to memorize, because these are the 4 phyla that keep showing up in all the human samples and mice. A lot of what they've done, they start out with mice or rats, and they're the same way. These 4 phyla dominate them as well. So Bacteroidetes and Firmicutes, Proteobacteria, and Actinobacteria. And it's kind of interesting, all these 4, initially a lot of the work was done with microbes in the gastrointestinal tract. But now they know that these 4 are also dominating the skin environment too. So it's both external and internal that we have these 4 phyla. And there's others too, but these are the dominant ones that we see a lot of members from these 4 phyla, lots of different species. So you can see 2 of them are gram-positive and 2 of them are gram-negative. And like I said, you are each unique. There are some unique differences that now with the better technology, they can pick up on those species that that are different for each person. So this is kind of a nice table trying to get across the idea of progression of the microbes from birth into several years out. And in some of these studies, they're also looking at breastfed versus formula fed. They can see some differences based on that variable as well. In this particular study that they published, they were showing a shift in the viral if it was breastfed or milk-fed babies that were being sampled. But you can see initially there's low species diversity, but then as the baby keeps growing, you start, and it makes sense. As long as the baby's not living in a bubble, you will get more diversity of species colonizing both external and internal the infant. We'll talk more about butyrate. That plays a very important role in your health. And then the toddler years. So this is one of the earlier studies where they were trying to get a handle on what's going on with human babies falling from the initial birth through a few years of growth. Here's another study that was published. 37 adults that were tested 5 years. 60 to 70% of the strains remained unchanged over the course of the study. And the most stable members of the microbiome tend to be the most So they're starting to look at this idea of those four phyla. You know, do they differ in terms of how easily can you manipulate them and shift their populations? So they're looking at perturbation. And again, you know, think about how would you mess with the population, especially of the gastrointestinal tract. It largely has to do with what you choose to eat, and that can make a profound difference on the microbes in your gastrointestinal tract. So they've been looking at skin microbiome as well, and this is, I like this picture because it's pretty easy to see that depending on which part of the body, well, there's samples lung as well. They're not just sticking. Actually, this is a bunch of different spots. So you can see that with the different colors on the pie charts, there's definite differences depending on the body site. And so they're trying to map all this out and get a better handle on it. But you can see, you know, lung tissue, a lot of people thought lung tissue was sterile unless you were sick with some pathogen, and they know that be not the case. There are microbes in low population levels in your lungs. So let's talk a little bit about some of the terminology that you see in this type of research. So they often refer to resident, they'll say, oh, the resident microflora. That's kind of what we're studying here for this section of the class. It's the ones that are, you know, they persist over time. They're not just ephemeral, there and gone within a day or so. That's more the transient idea where you pick them up from some environmental surface typically, and they persist with you for a little while, and then they die off because it's not, you know, your own microbiome can often outcompete them, or they're just not suited to wherever they landed on you. And then the opportunists, I won't talk too much about the opportunists. That's more of the pathogens that can get in or on you and cause problems. And so I'll mention a little bit about those, but not much. We're going to focus on the normal microbes that hang out with you long term, and we're now finding are doing all kinds of good things for you. And so we are going to focus mostly on the resident microflora for this section. So as we work through and think about the different body parts, again, just trying to I guess, reinforce the idea that they're everywhere pretty much. And so here you're looking at skin surface. And you know, I think you probably all are aware that you are shedding skin all the time. And so the microbes persist in a little bit lower down. So your skin secretes enough oils and other organic acids, and so they like to hang out. They really like hair follicles. They like sweat gland areas, sebaceous glands. So they can be found a little deeper down. They're not just at the surface. And, you know, again, there's some bacteria that are better adapted to your skin. Your skin provides, you know, slightly acidic pH. And then you have some different nutrients that are really putting a selection pressure for certain microbes to adapt to. Others that land there may not be able to make it and persist. This is nice too. This is showing, again, with the different skin, whether it's sebaceous, moist, or dry. So these three bar graphs on the right, you can see the different colors and the different proportions of these colors. It's a very nice visual showing that it depends which body part you're looking at. You have to remember the size scale difference between your body and the microbe. They can get into all of these different areas, millimeter or so of your skin, but you can see different ecosystems depending on what body part you're looking at. And on the left, the pie chart looking at the different phylum, and you can see with the skin, gram-positive. 3/4 of the pie chart. So they dominate your skin. If you start to go a little bit more internal, so the oral and nasal cavities, you have a lot of epithelial cells on the surface that can get well colonized. And you can even see like species, Neisseria species, and like Neisseria gonorrhea is famous, but there's non-pathogenic Neisseria that can hang out in the nose throat area. You have some gram-positives like Streptococcus and Lactobacillus that can also be present early on. And so again, when you think about the oral microbiome, there's a lot of more research going into this now, more grant funding being thrown at oral microbiome, because they're starting to realize how important your oral health is to the rest of your body, including your brain. There's some interesting hypotheses about certain brain diseases like Alzheimer's may be linked to certain poor outcomes with oral hygiene. We'll talk about that later. But anyways, so here we're looking at this idea of what happens when you have no teeth in the mouth, and then once teeth start to emerge, what begins to happen. And again, we see this succession of microbes because you're providing a different type of environment. Initially, a lot of the surface in the human infant mouth is more aerobic than anaerobic. And then as you get teeth, well, now you have all these pockets and cracks and crevices where anaerobic conditions can be set up quite quickly, where the teeth and gum line up, especially. And so then you start seeing a shift where you can get facultative anaerobes in there that build up in population, which then can create an environment for anaerobes to come in and set up camp and live there. And so Prevotella and Fusobacterium are ones that are shifting their facultative anaerobes. So you start to see this kind of aerobic, facultative to anaerobic, and then you a mix of all three by the whole time the mouth is fully erupted with teeth. But basically, you see the succession where the first colonizers that get in there start to create environments that allow for other microbes to get in. And once the facultative anaerobes can get in, they can create anaerobic environments. They scarf up all the oxygen preferentially, and that leaves anaerobic state that the other ones now can come in. And so there's a lot of really nice studies looking at succession. And so, you hear a lot about some of the microbes that can cause like dental plaque and dental caries, but there's plenty of other microbes in your mouth that aren't harming you at all. And, every time you brush your teeth, you're bringing down the populations of microbes in your mouth, but you're never totally getting rid of them. And so there's even studies showing that if you gargle too much with alcohol-based oral rinses, that's actually a bad thing. They say that you should, every once in a while it's fine, but doing that, I think the study I read, they said 2 gargling events per day was actually a poorer outcome for your oral populations. It wasn't doing them a good thing. It was actually causing problems. So you need to think about that a little bit. Here again, just trying to get across that you have a mix of interesting microbes that are hanging out in the nose throat area. Certainly ones that can get out of balance and start causing disease. If you have poor oral hygiene habits, then yeah, you can start to get the bad guys gaining a foothold too strongly, for sure. But you have a lot of microbes in there that are capable of keeping the bad ones in check, as long as you're taking care of your mouth correctly. The tongue, especially, is they've done some really interesting research on the tongue. It's just chock full of microbes. And the last time I went to my dentist, who actually is a former NMU student, he graduated from our biology department. He took my plant phys class, and now he's my dentist. It's kind of fun. I'd like to chat about the independent research project that he did in that class. He still remembers it. So, but anyways, I was asking him, I was picking his brain. I'm like, so I'm teaching about the oral microbiome in Gen Micro. I'm thrilled to hear that I spend a little bit of time on that in class. I said, is it better to do a tongue, those tongue scraping things that you can buy, I said, is it better to scrape your tongue or brush your tongue? Because I've heard people, are saying, oh no, do this, I'll do that. And he said, basically, it doesn't matter. Either one is sufficient. And now I found a tool that does both. It's like a scraper and a brush in one tool. So I'm like, oh, I'll try that. So again, when you're scraping your tongue, you are scraping off some of the microbes, but you can't get rid of them all. You're not going to do that. And you don't want to get rid of them all. You just want to keep the balance of the population in check. Every morning when you wake up and you have the biofilm across your teeth, that's like, okay, yeah, you need to brush your teeth to get that population back in check. Here's a nice study that they did. This is the tongue surface, and they have color-coded different groups of bacteria that they were studying. So let's see if I can enlarge that a little bit. So the actinomyces are the red ones. Streptococcus are green. And then some presented, they're grouping up in clusters and stripes. It suggests growth of the community outward from the central core. So they think that the growth is separating. So they're really getting into it, looking at, where are these microbes hanging out exactly on the tongue. So really interesting research going on with that. That's the paper that image was taken from. So a lot more diversity there than initially thought. And definitely a successional process over time when you think about pre-teeth and then the eruption of teeth. So we have certain enzymes that also help keep populations in check. We have lysozyme in our saliva and lactoperoxidase in our saliva. And that does, lysozyme is really good at, well, they both are at breaking down the cell wall and lysing cells. So you have that going on. And certainly you have the shifting of aerobic bacteria into more anaerobe showing up as teeth emerge in your mouth. And then every morning you do have a biofilm. And then if that gets out of control, it can lead to dental plaque, which is harder to scrape off. Just brushing won't do it. You get that kind of calcified stuff showing up on your teeth that need to be scraped away by the dentist. And so also lactic acid bacteria like to hang out in your mouth. And at those, you want to make sure your population levels are low enough because that can eat away at your enamel and cause problems. So you need to be careful about that. And then this image is looking at the tooth and trying to show how the gum, comes up and supports the tooth. And so you have a lot of areas, all in that pocket against the tooth below the gum there. You have lots of microbes that can hang out there and live their life. So yeah, think about that next time you're kissing someone. You're swapping a lot of microbes. So the other looking at is dental plaque or biofilm growth over time. And so I think this is the study where they didn't have the person brush their teeth for 10 days. And then I can't imagine, but paid me enough, maybe I do it. But anyways, so you can see significant biofilm formation. These are longitudinal sections through the biofilm. So, I mean, it's just chock full of microbes, all of that. Those are cells in the biofilm. Here they've done some studies where they had artificial teeth, kind of like an artificial mouth system in the lab that they were studying. And they were just trying to get a handle on how quickly do the microbes attach, how long before biofilm formation. So they're getting some really good data, looking at the whole process. This is from another published study, just again, trying to show the diversity. Biofilms especially on the tooth surface is fairly common. It happens very quickly. So if you're not drinking, and that's why every morning you get that, because you a whole night of not drinking or eating, and so things have a chance to build up because everything stays stationary. You don't have a bunch of fluids going over your teeth during the night. So this is the paper that was published back in 2019, and there's still several groups that are following up on this. So this was taken from the New Scientist magazine, summarizing data experiments that have been done. And so they were summarizing this paper where they were looking at Porphyromonas gingivalis, which is heavily involved in gum disease. It gets out of check. And what they were looking at is the link of that. And they're saying that it invades the brain from the mouth. So somehow you get that transfer if the population gets high enough. And so they were tracking, they had known for quite a while that Alzheimer's patients tend to have poor oral health and that they have issues with their oral microbiome. And then they kind of stumbled on this idea when they were, what's the word when the postmortem exam, when they are looking at the brain tissue, they noticed they were looking at the brain tissue for other things, and they kept like, what is this? What is this? And then they looked more carefully, and they're like, there's bacteria in the brain tissue. And they were quite stunned by that. Well, then they started looking for it with all those postmortem exams that they were doing, and they were finding it. And so that's what led them to really start thinking about this more carefully, and they were able to identify this Porphyromonas showing up. And so then they did mice studies where they were trying to get this to happen in mice, and they were showing that it can, they engineered mice to have Alzheimer's, and it caused Alzheimer's-like brain inflammation in the healthy mice. And so again, it's very common, of course, with this kind of study, going into mice and rats and doing it there is a whole lot easier. But you have to remember, mice and rats are not humans, but they often are used. And then there's this study that came out where they're like, well, but wait a minute. And so this one is also the idea that they were finding accidentally microbes showing up in their studies, which they didn't expect. And they were actually looking for schizophrenia. They were trying to track down what's going on with that and looking at postmortem brains with that. And again, found that there's microbes and they don't seem to be doing anything dangerous to the brain in any. And so it's still, you have these different studies showing two very different ideas. One could be leading to the pathology of Alzheimer's. The other one is showing, doesn't seem to be a problem. And so this is probably another really hot topic right now, where they're looking at this very, very carefully, because we always used to think the brain was pretty sterile, that there was no way to get any microbes in there. And now they're starting to question that idea. So 34, in this particular study, it says 34 human brains and found bacteria in every single brain. Okay. So brain tissue is on the agenda for what's going on there. They're really trying to track that down. So again, most of what you see in the mouth and nose and throat area are not something to be worried about. It's just a handful of microbes that cause problems. But some of those can cause some serious problems in other parts of the body. So like the Alzheimer idea, that's very intriguing. That's still a story that needs to be fleshed out more. Could microbes migrate to the brain problem? Here you're looking at pictures of endocarditis where the oral microbes migrated into the heart, protective heart tissue, and were able to cause issues. And so if you have oral surgery in particular, it's of concern. They often, I think most dentists prescribe antibiotics for you before you have oral surgery, primarily for this reason. They're trying to stop microbes from migrating out of your mouth because, during oral surgery, you're going to have bleeding and you're going to have the opportunity wounds and all of that. So that increases the chance the microbes will escape your mouth and get into your bloodstream more readily and could end up somewhere where they shouldn't be. And so this is something that they've known about for a while, that they need to be careful before they start doing oral surgery. Okay. Respiratory tract, when I took microbiology, they said it was sterile unless you were sick with a pathogen. Now they looked at it more carefully. The upper respiratory tract is well colonized, not abundantly, but well colonized by microbes. They're there. And then the lower, they are also finding that they used to say, oh, no, that's sterile. And now they're finding, oh, no, there are actually non-pathogenic microbes that hang out there. Certainly, you can have pathogens, there's cystic fibrosis. It usually starts in the lower lobes of the lung and works its way through the whole tissue. But now we know that there's normal microbial colonization and they are not causing problems. So they're finding Capitella, Streptococcus, Salmonella, and so, we have a lot of cilia lining our cells in the lungs, and that certainly helps to keep the populations in check because it sweeps things. The escalator, the mucociliary escalator, I like that phrase. And so it sweeps up. And so that's one way for our body to do things in check, make sure things aren't overpopulating. So the respiratory tract is certainly capable of being colonized. The bladder is usually sterile, that at least so far the reports I've read, they're still saying that, unless you have a bladder infection, of course. The urethra, however, is usually constantly colonized by good and bad, depending on what's going on. And so the opportunists, of course, can get in there and urinary tract infections. But you normally have a microbiome colonizing the urethra. So have you ever heard of bladder stones? I don't know. I haven't talked to any human that's ever had bladder stones, but my dog just had surgery not too long ago for bladder stones. And I didn't even know that was a thing, and that dogs could get that. So the first type of bladder stones were due to a urinary tract infection. So apparently the microbes in the urethra made it up into the bladder, and they excreted urease enzyme, which made her urine, it acted on the urea of the urine, and it allowed for stone formation to form. And so it was just the presence of these, it's usually Staphylococcus and Proteus species in dogs that will cause bladder stones. And so because they had the urease enzyme, it allowed for stone formation. And when the vet showed me the x-ray of her bladder, it's like it was chock full of stones. I mean, I don't know how she fit any urine in. It was all these stones, some big, some small. I was wondering why she wasn't dogs have a good way of hiding pain, but it's like, wow, I felt horrible. So be careful if you have pets. Make sure that you are, I didn't know she had a urinary tract infection. She had no obvious symptoms. So it's something now that I have to keep on top of. So vaginal canal certainly is well colonized. And again, this can shift over the life of the female. Slightly acidic as an adult, but and so with slightly acidic conditions, lactobacillus are very popular, some of those species. And then Candida is a yeast, and that, remember, fungi like slightly acidic. So that makes sense that they're there. And so normally vaginal tracts have Candida. And I know a lot of people are like, well, but that's a yeast infection. Well, yeah, if it gets out of control and the populations go up too far, then you'll get a yeast infection. But normally Candida is just there. and it's fine. It's not going to be a problem. Very young females or older females, you tend to see a different, slightly alkaline pH, and that will support different microbes. And so you'll see different microbes popping up during those phases of life. And so again, it just pH can strongly influence the vaginal tract microbiome. And then, of course, the menstrual cycle also changes the population numbers and composition as well. So that every month when that's going on, that happens too. And then sometimes if you have antibiotics, if you take antibiotics for something, it doesn't have to have anything to do with vaginal health. If you just are sick with something else and you start taking antibiotics, it can trigger a yeast infection because it kills off some beneficial microbes in the vaginal tract and allows Candida to get more of a foothold. And so essentially what it's doing is it's reducing competition, and Candida starts to have more freedom to nutrients, more freedom of space, and it starts to overgrow and cause a yeast infection. So another reason to be very cautious about antibiotic therapy. Sometimes you absolutely have to do it, but sometimes you might not have to. Okay, so the next topic we're going to get into is zooming in on the gastrointestinal tract, and we'll spend a good chunk of time over the next several days or lecture periods on the gastrointestinal tract. So, but we'll stop there for today.